Thank you very much, Tim, for sharing that. My name is Ulrike Potzer. I'm a virologist working on ICE-HBV, I don't know, since many, many years. I'm from Munich, Germany. And it's my pleasure to introduce the next speaker, who's also from Germany. It's Florian from Bömmel. Florian is a hepatologist, gastroenterologist, currently the deputy head of the Department of Gastroenterology and Hepatology at Leipzig University. And Florian started very early on to study the hepatitis B virus RNA as a biomarker in the serum. And at the beginning, nobody believed him so really strongly. But I think now people, Florian, believe you, and we're looking very much forward to your talk.
Thank you, Ulla. Can everybody see my slide? Yes, I think so. So thank you for the nice introduction, and hello to everybody present in this meeting. What is HBV RNA is the title of the talk I was asked to give today, and I want to start up front with a little scheme here about HBV gene expression. And here you can see the genes of the hepatitis B virus, which are probably not below, and you can see that there is a couple of messenger RNAs that are needed to produce all those proteins. The longest is a pre-core RNA, then the pre-gene RNA, then we have two RNAs for the surface antigen and one for the X gene. And this is basically what we are talking about when we talk about HBV RNA, but there is more to it. There are spliced variants of the hepatitis B virus. And this is a figure that comes from the first group I was working in Hans Wild's group in Hamburg, and I had the pleasure to start working on HBV RNA at the time. And here the colleagues were looking at all described and possible spliced forms of HBV RNA. And there is a number of spliced RNA variants. They are spliced at different sites, and then they're used together by the excretion process of HBV RNA. I just want to show you two studies that for now one comes in, and here research is called CAX from the bio group. So I have added these two here to the life cycle. So HBV RNA is probably present in virion-like particles. There is evidence that there are naked capsids that contain HBV RNA, but I don't think this issue has been solved to the end. But yeah. This is what we know for now. In serum, we have different HBV RNA species. So we have the messenger RNAs and the pre-genomic RNA that I have shown you. We have spliced variants, and we have different RNAs with different splicing or with different length at the 3' end. So there is a truncated and a full-length form, which we do not know a lot of, but this is a summary of the RNA forms that have been described to be present in serum. So although there are now, and I'll show you this later, there are now many papers that claim to look at pre-genomic RNA, and I'm not doubting their methodology, but it has to be underlined that there's probably a mixture or a quasi-species of HBV RNA present in the serum. And it has not been shown whether this is different in different clinical situations, for example, in acute and chronic phases or in HCC development, or whether this has something to do with integration of HBV or with a response to treatment. And this is what I think a very interesting point. So extracellular serum HBV RNA contains pregenomic and spliced RNA variants. I mean, this has been shown. This is these are two different studies here that have that could show this. So here we could see that here spliced variants were present in the serum in treated patients with nucleoside analogs and in naive patients, and here in cell supernatant. So it is very likely that the spliced RNA is indeed present in patients. What is HBV RNA is the title of my talk. And one question is, how is it related to intrahepatic HBV RNA and to cccDNA? And this was answered by a very large and very sophisticated study by Wang and colleagues that was published in the Journal of Hepatology in 2018. And they were looking into cccDNA sequences, intrahepatic RNA sequences, and extrahepatic RNA sequences. And they could show that there is a large homology across all three species. So what we see in the extrahepatic compartment is very much the same of what is present in the cytosol, and this is very similar to the cccDNA DNA sequence. Why is this important? It is important because these messenger RNAs, they surely undergo regulations, and it could be that they are selected by some processes that we do not know, but apparently they are a very reliable marker of cccDNA. And they are distributed equally in the liver across the different functional units of the liver. So in the lobal, in the central vein, in the portal vein. Here in this study here, around 50% of hepatocytes were positive for HBV RNA, and so they can be found in all compartments of the liver. How can HBV RNA in serum be measured? And there are different strategies, and I'll show you one strategy that was the first to be published in 1996. And this is by Kirk and colleagues, and they have developed a race real-time PCR targeting the oligo-DT, the poly-I tail that all those messenger and the pregenomic RNA have. And I think this was a very smart approach. They took one primer here and another primer on the HBV genome, so they could evade cross contamination with the HBV DNA without any DNA's digestion or any removal of DNA. And this is actually a methodology that we have also adapted for our studies and that we find very robust and that we recommend to others. In this very early study, HBV RNA could be found in serum, in HBV positive PBMCs, and in mice here. So there's a variety of methodologies that have been presented in the meantime. So there is a real-time PCR, there's a digital droplet PCR that has the advantage of a little bit of a lower limit of quantification, but it has to be said that the real-time PCR can be, well, the limit of quantification is variable, of course. It can be adapted to the need of the study. There's the 3' and race-based PCR with oligo-DT primers that does not include DNA's digestion, DNA removal. There's a quantigen assay, and there are indirect measures over pre-core and core regions that have been published. So almost everything based on PCR. Here, the question is now, how can these methods be compared? And there are only few papers, I think only one that shows a neat comparison of two different methods. And this is a study published by Butler et al. And they showed how an assay that detects simultaneously HBV RNA fragments over the S and the X gene that was developed by Abbott compares to the race PCR primer based assay. And you can see that The results are quite homogeneous in a certain range here. However, in the upper range, they tend to be more diverse. And I wish that this study would be repeated in a larger patient or sample cohort. That would be very helpful to understand the performance of the two different assays. But at least they seem to be comparable over a very large range. So how is serum HBV RNA associated with other serum markers? First of all, serum HBV RNA and intrahepatic HBV RNA are significantly lower in e-antigen negative infections and inactive carriers. And this has been shown in the study from Wang that I quoted earlier. And here you can see that the inactive carriers have undetectable or very low HBV RNA levels as compared to e-antigen positive disease or e-antigen negative disease. And this is holds true for serum and intrahepatic HBV RNA. Here it was shown, this is only one of a couple of studies that showed that there is a strong correlation between serum HBV RNA, which was named pregenomic RNA in this case here, and intrahepatic CCC DNA here and intrahepatic pregenomic RNA there. This is a study that was done in a very large patient cohort, and here the authors were looking at HBV RNA levels in comparison to HBV DNA, to quantitative S antigen and E antigen, and they found the correlations were different across the genotypes that were included. Here you can see the genotypes, and each genotype has a different line here that shows the Pearson correlation. So there are certain differences across genotype, and importantly, there are differences in e-antigen positive and negative patients. So in e-antigen positive patients, there is a similar correlation of HBV DNA and RNA as in the negatives, but S antigen correlations are completely different. There hardly seems to be any or only a very weak correlation of S antigen and HBV RNA in the e-antigen negatives patients. And one could ask where this comes from, but as we are increasingly understand S antigen, HBs antigen in e-antigen negative patients predominantly is derived from integrated HBV genome. So if this is true in this patient cohort here, then HBV RNA and S antigen may have different sources. And this is very interesting because all of them are products of cccDNA replication in e-antigen positive patients, but this may not be the case in e-antigen negatives, and it may be helpful to distinguish between S antigen and other replication markers here. So the significance of serum HBV DNA in the clinic, is it a good tool to monitor cccDNA activity? And this was proposed by the Abbott group here, Butler et al., and they show that during nuke-unlike treatment, you can suppress HBV DNA, but the RNA level remains detectable and finally comes down to undetectable level. And this could show a decreasing of cccDNA activity, or maybe of the cccDNA copies that are in the cells. And this is a very, very interesting hypothesis. If this is true, then we have a marker in our hand that gives us information about HBV replication in patients under nuke treatment that have suppressed HBV DNA here. And this is indeed what we could show when we published our first results. We could show that HBV RNA levels strongly differ between patients who have e-antigen seroconversion in the future from here. So they have very early on a decrease of HBV RNA here in orange to undetectable levels as compared to those patients who do not achieve e-antigen seroconversions. They have continuously detectable and intermediately high HBV RNA levels. So this is a huge difference here. So HBV RNA is an early marker for e-antigen seroconversion. Why is that so? I think this question is still open. It can be that cccDNA activity is becoming shut down prior to e-antigen loss in those patients here. It can also be that the molecules containing HBV RNA are somehow being removed from the serum by an immunologic event. So although this seems to be a reflection of cccDNA activity. In my opinion, it is not clear whether we see a decrease in cccDNA activity here or just a decrease in the particles containing HBV RNA. And this is something that needs to be researched in the future, I think. We have also looked into a cohort that was provided by Gilead Sciences from the prospective randomized study, and we have looked at different markers here: HBs antigen, correlated antigen, HBV RNA, eantigen, and HBV DNA in patients who have S antigen loss. Here, these are the blue ones, and the purple ones S antigen positive but E antigen loss, and the gray ones have either form of serologic response. And we can show that those three groups have very distinct kinetics of those different markers here. And if we look at HBV RNA, there's a very early significant difference across the three groups. So it can be, but this needs to be shown in larger cohorts, that HBV RNA in serum is also a very early marker for S antigen loss. It is a marker for the relapse of HBV.
Yep. Sorry to interrupt you, but I think we need to come to an end.
Okay, I will be very fast, I promise. It is a marker for the relapse after stopping nucleoside analog treatment. Here is a retrospective analysis of the finite study, and it is a marker of histopathology. And it is a marker that is often used in novel treatments. However, we do not have an endpoint here, but we see it as a marker that responds. And this is the answer to the question. HBV RNA questions for research. We need to define the composition of serum RNA. This is still open. The role of the truncated RNA is open. We need to define RNA as a clinical biomarker in novel prospective trials. But today we can already say it has the potential to become a marker for HBV infections, and it is probably a marker of cccDNA activity and of the existence of HBV RNA containing particles. Thank you very much for your attention, and forgive me for being too long.
Thank you very much, Florian. Sorry for interrupting here, but there was a lot of questions in the chat, and I really wanted to allow some of the questions to be answered. One question is, do you think the HBV RNA can also stem from the integrated HBV genome, or is that mainly from cccDNA? And if it is from the integrate, would that be a different form, or is it exactly the same?
So a very good question, and actually that's a question our research has focused on for some time now. So if integrated HBV genome is the source of HBV proteins, there needs to be HBV RNA that communicates between the integrated cccDNA and this protein, which is necessary for the translation. So there is obviously HBV RNA that comes from integrated HBV genome. The question is, can we find this in serum, and can we distinguish it from other HBV RNA forms? To date, we cannot. But I think it is probably possible. The more we learn about integration, the more we will be able to find specific HBV RNA species that are associated with it.
Then there is a question from Akira Nishio. Is HBV RNA reduced with nuke treatment, or maybe I can add on any other antiviral treatment?
So if we treat with nukes, we see a reduction in HBV RNA, a fast reduction in patients who have serologic response in the future. If we treat for a long time, and we did a very interesting analysis with Pietro Lampertico, we were looking into patients treated for more than 15 years with nukes, and we see a decrease in most patients to undetectable levels. So in the very long-term treatment, most patients become undetectable by HBV DNA by currently available assays.
What about newer therapies like CPANs?
Yeah, and in these treatments, HBV RNA levels are also looked at, and they become undetectable very early on. But again, in this treatment, when you interrupt the capsid formation itself, you're obviously tackling the transporter for HBV RNA. So it may have a different value in this treatment as it has with nuke treatment.
Mm-hmm. And maybe a last question, how much of spliced RNA do you detect in the serum?
It's a very good question. So we have, I can say it frankly, we have difficulties to detect it in our group, but it has repeatedly been detected. So I believe that it is there. Actually, there's no large-scale study today that looks at different HBV RNA species in different clinical situations. And this is a study that is, in my understanding, urgently warranted, because I cannot answer the question. I think it's there, but it's hard to detect.
And the very last question, there is no standardization of assays so far. Is there any developments in diagnostic assays? I mean, I know that you're involved in some of the studies.
Yeah, so there is a diagnostic assay that is available for research by Abbott, and there is one that is in development by Roche. Both have a little bit different technologies, but I think they will deliver comparable results. And yeah, we will see how soon they will be available for clinical practice.
Okay, thank you very much, Florian. That was a great talk and obviously of great interest to the community.
Thank you for the
Thank you very much.